Welcome to the Longevity Papers podcast.
Great to be here.
This week, we're doing a deep dive into a batch of sources straight from longevitypapers.com. These are curated specifically for, well, for you listening, the biotech researchers out there.
That's right. The goal is really to cut through the noise, get to the heart of what these new papers are saying.
And figure out what they actually mean for, moving the needle on extending healthy human life. That big goal, that order of magnitude leap we're all interested in.
Definitely. And we should be upfront. We're looking at some bleeding edge stuff here.
Yeah, a lot of these sources, especially the ones from Bjork's pronounced bioarchive folks, they're preprints.
Meaning they haven't necessarily gone through the full peer review gauntlet yet. So a healthy dose of skepticism is definitely warranted. We need that truth-seeking eye.
Absolutely. And this week, it feels like a good time for a check-in on the start of the field, based on these papers anyway. Are we seeing, huge paradigm-shifting breakthroughs?
Or is it more like solid incremental progress? And, well, based on the sources you provided, the honest take is it seems mostly like the latter this week.
Right. The executive summary for this batch puts it pretty bluntly. It says there's about a 95% probability that these papers represent statistical noise or just marginal improvements relative to that really ambitious goal, adding a decade or more to life.
Which sounds a bit harsh, maybe, but it's an important perspective. That said, even incremental steps or developing powerful new tools, they're crucial for the whole journey.
Exactly. They build the foundation. So let's unpack what we do have, starting with a paper that takes us deep into the cell, looking at lysosomes and hinting at something, well, kind of wild. Transgenerational effects.
Okay, yeah. This first one is titled, Lysosomes Signal Through Epigenome to Regulate Longevity Across Generations. It's up on Bioarchive, dated May 24th, 2025. Authors are Zhang Deng Wang from HHMI Genelia Research Campus.
Lysosomes, I usually think of them as just the cell's, recycling center, garbage disposal. What did they find that's relevant to longevity?
Well, that's what's so fascinating. Phil, these lysosomal metabolic pathways aren't just passive. They're actively signaling to the epigenome, those modifications on or around DNA that can be inherited.
Okay.
And this signaling seems to regulate longevity, not just in the organism itself, but potentially across generations. So this work was done in C. elegans, the roundworm.
Wow, okay. Transgenerational longevity linked to cell garbage disposals and DNA modifications. That sounds complex. How do they think it works? What's the proposed mechanism?
So they found that tweaking certain lysosomal pathways, like activating lysosomal AMPK, or maybe reducing mTOR signaling there, it boosted the expression of a specific histone variant, H3.3, and it increased a particular modification, H3K79 methylation.
Okay, histone modifications, epigenetics 101. But what's the transgenerational part?
Right, this is the really novel bit. They identified a potential mechanism where this H3.3 protein carrying that epigenetic info is actually transported from the worm's body tissues, the soma, to the germline, the reproductive cells.
Whoa, soma to germline.
Yeah, and combined with a specific enzyme in the germline that does that H3K79 methylation, this seems to be how that longevity signal gets passed down.
Okay, soma to germline transport of epigenetic information. I mean, that challenges some fundamental biological ideas, doesn't it, if it holds up beyond worms?
Exactly. And that's where the skepticism has to come in, right?
For sure. What are the main points?
Well, the biggest one is the model organism itself. C. elegans is fantastic for discovery, no doubt. But translating findings from worms directly to mammals, let alone humans, it rarely works out perfectly.
The pathways might be different, the whole epigenetic landscape.
Precisely. And this specific soma to germline transport thing could easily be worm-specific. Plus, the exact role of that H3K79 methylation in human aging isn't really nailed down yet.
So high novelty, potentially high impact if it translates, but big ifs. What experiments do they suggest next?
Well, they suggest the logical steps. First, directly test if overexpressing H3.3 in mouse oocytes, the egg cells can trigger similar effect across generations. That's a key test for conservation.
Makes sense.
And second, try to develop drugs, activators specifically for lysosomal AMPK that work in mammalian systems. See if manipulating that pathway has similar effects outside of worms.
Right. So given the novelty, but also that big translational gap, how does the source rate the impact potential here?
This one gets 4 stars out of 5. And the description captures it. High risk, high reward, especially for efforts in epigenetic reprogramming.
So huge potential if the mechanism holds up in mammals, but major unknowns right now. It's a really interesting discovery, opens up new ways of thinking, but yeah, the path to a human therapy isn't exactly clear from this paper alone.
Not yet, anyway.
Okay, let's shift gears then from worm epigenetics to something quite different, modulating blood factors. And importantly, this next one was actually studied in humans.
Yes, this is the paper titled Multi-Omics Analysis Reveals Biomarkers That Contribute to Biological Age Rejuvenation in Response to Single-Blinded Randomized Placebo-Controlled Therapeutic Plasma Exchange. Published on PubMed, May 27, 2025. Big author list: Fuentealba, Kiprov, Schneider, among others, out of the Buck Institute for Research on Aging.
The Buck Institute, yeah, definitely a major player. So, therapeutic plasma exchange, TPE, that's a clinical procedure, isn't it? Like filtering blood plasma.
That's exactly what it is. And the core finding here is pretty direct. They report that TPE seemed to rejuvenate biological age markers in a group of healthy adults over 50.
Rejuvenate biological age markers.
Yeah, and that's relevant because these biological age clocks, they're thought to reflect underlying aging processes, things linked to health span and potentially lifespan.
And this is in humans. That feels like a significant step up from the worm study in terms of immediate relevance.
It absolutely is. And the paper makes a point of saying this is the first human study showing this kind of rejuvenation across multiple omics. So looking at epigenetic clocks, but also the proteome and metabolome just by modulating blood plasma factors.
Interesting. Did they look at how often the TPE was done?
They did. They tested different frequencies, and it looked like monthly TPE had the strongest effect on these markers. So maybe a dose-response relationship there. And importantly, they reported it was safe, well-tolerated in this group.
Okay, that sounds promising. A tangible intervention. But where does the skepticism come in for this one? What are the caveats?
There are a few key ones. First, the study size was pretty small. Only 42 people randomized across the different groups, including the placebo arm.
Right, small numbers always make you cautious.
Exactly. Even with the placebo control, things like regression to the mean can be a factor in smaller studies. Also, a potential confounder.
Yeah.
Some of the treatment groups received IVH, that's intravenous immunoglobulin, along with the TPE.
Ah, so it muddies the waters a bit. Hard to separate the TPE effect from the IVE effect.
Precisely. It makes it difficult to say definitively that the effects are just from removing plasma factors via TPE. Some could be from adding beneficial factors back with the IV.
Okay, so human data, but small scale, some confounding factors. What are the suggested next steps then?
Well, the obvious one is larger randomized controlled trials. Definitely needed. And crucially, these future trials need to look beyond just the molecular markers.
You mean look at actual function?
Yes. We need to see if this rejuvenation, shown in biological age markers, actually translates into real-world improvements in health span. Things like measuring physical function VO2 max, grip strength, or cognitive function, or even tracking incidence of age-related diseases.
That makes sense. Markers are great, but function is what matters.
Right? And the other big goal they mentioned is trying to isolate the specific factors in the plasma, the ones being removed or added, that are actually responsible for the effects. That's the key to developing more targeted therapies that wouldn't require doing the whole plasma exchange procedure.
Got it. So how does the impact potential rate for this TPE study?
This one gets 3 stars. One or, and the description is clinically actionable, but incremental.
Actionable because TPE exists.
Exactly. It's a procedure already available, but incremental because while it shows some promise with these markers, the current evidence doesn't really suggest it's, the big answer for adding decades to life right now.
So a promising avenue may be leading to future therapies based on specific factors, but the effect shown so far is modest and based on markers.
That sums it up well. Needs more work.
Okay. Moving on from blood factors to another key hallmark of aging, cellular senescence. And this next paper seems to tackle a technical problem in studying it.
That's right. This one is improving cellular senescence detection with weak gene markers in single-cell RNA-seq via iterative imputation. From Biorex again, May 29, 2025. Authors include Zhu, Zhang, Zhu from Southeast University.
Cellular senescence, those dormant cells that build up and cause problems as we age. What's the challenge they're trying to solve here?
The big challenge, especially now that people are using powerful single-cell techniques like single-cell RNA sequencing, is just accurately finding which individual cells are actually senescent.
Why is that hard? Don't we have markers?
We do, like P16 or P21, but the problem is they can be expressed at really low levels or inconsistently across different cell types or tissues. It makes getting a clear picture really difficult, especially at the single-cell level. It's a genuine technical bottleneck holding back research.
Okay, so it's hard to reliably spot them, and they've developed a tool to help.
Exactly. They built a computational framework they call ICE, stands for Iterative Imputation-Based Cell Enrichment. The key idea is that this tool significantly boosts the accuracy of finding senescent cells, especially when you're relying on those gene markers that might be weak or spotty in single-cell data.
How much better is it?
They're reporting a pretty dramatic 98% accuracy improvement in their tests, and they validated it in systems relevant to aging, looking at microglia, immune cells in the brain, in an Alzheimer's model, and also beta cells in the pancreas during aging.
Wow, 98% improvement. So ICE sounds like a much, much better magnifying glass for finding these senescent cells. But it's still just a tool, right? It doesn't do anything about them.
Precisely. That's the main point for skepticism, or rather, perspective. ICE is a computational tool. It's potentially incredibly valuable for researchers helping map senescent cells better, understand their differences, maybe find new types.
And it doesn't remove them or treat diseases.
Correct. It improves our ability to study senescence,
Yeah.
which is vital, absolutely critical groundwork. But it's not an intervention itself. And it still ultimately relies on the biological markers we have, which, as we said, might have their own limitations.
Okay. So what kind of new experiments does having a better tool like ICE actually enable?
It really opens things up. Researchers can now apply this to much more complex data sets. Think about human biopsy samples from aged people or diseased tissues, where finding senescent cells accurately has been super challenging.
And maybe evaluating therapies.
Yes, that too. It could allow for a much better assessment of senolytics, those drugs designed to clear senescent cells. With ICE, you could potentially get a much more precise measure of whether a treatment is actually working, whether it's reducing the senescent cell load in a tissue.
So quite impactful for the research process itself. What's the impact rating?
This one also gets 4 stars, but the description is field advancing for senescence research.
Right, emphasizing its role as a tool.
Exactly. While it's not a direct therapy, having a tool that significantly improves how we study a key hallmark of aging is hugely impactful for the entire research field. It tackles a major technical hurdle lots of labs are struggling with. Better identification is just crucial, both for basic understanding and for checking if treatments work.
Makes sense. Okay, let's circle back to metabolism now, another central theme in aging, specifically looking at NAD+ and the enzyme CD38 that degrades it.
All right, paper number 4 is multiplex targeted spatial mass spectrometry imaging assays to monitor lipids and NAD+ metabolites in CD38 knockout mice exhibiting improved metabolism. Another Biowork for preprint, May 26, 2025, and also from the Buck Institute. Authors include Sherman, Bonds, Kumar.
CD38, yeah, it's pretty well known for chewing up NAD+ where and NAD+ declines with age. So what's new here? What did this study add?
Well, the core finding confirms what we generally know. Knocking out CD38 in mice leads to improved metabolic profiles. They saw changes in lipids and NAD+ metabolites. But the really exciting part, I think, from a technical standpoint, is how they looked at it.
Oh, so?
They used a new spatial metabolomics technique. It's called IPRM-PASEF. Basically, it lets them visualize and map specific molecules like lipids, NAD+ swerves, its metabolites directly within intact tissue samples, and with really impressive resolution.
So it's not just measuring the overall level in the liver, for example, but showing where in the liver cells or the tissue structure those molecules are.
Exactly that. It gives you subcellular or subtissue spatial detail. So while we already kind of knew CD38 was involved in age-related NAD+ decline and metabolism, this technique provides an unprecedented level of granular validation.
Can you give an example?
Sure. They can show, for instance, higher NAD+ levels and lower levels of ADPR, which is a byproduct of CD38 activity specifically localized within the liver cells, the hepatocytes, of those CD38 knockout mice.
Okay, that's a really powerful technical advance for studying metabolism right where it happens. What about the caveats, the translational questions?
Well, the main one, again, is that it's a mouse study. CD38 and NAD metabolism are conserved, sure, but mouse models don't always capture the full complexity of human aging or metabolic diseases.
Right, and anything specific about targeting CD38 in humans?
Yeah, there's the potential issue that completely knocking out or inhibiting CD38 systemically in humans might have knock-on effects, particularly with the immune system where CD38 plays roles.
Yeah.
That needs careful consideration for any potential therapy.
Okay, so what kind of follow-up experiments does this work point towards?
Well, they suggest using tissue-specific CD38 knockout models, maybe moving into primates, to get a better handle on translational potential
Yeah,
and maybe avoid those systemic effects.
makes sense. Target the tissue you want to affect.
Right. And they also propose using this cool spatial imaging technique to better understand how NAD plus precursors, things like NMN or NR that people take, actually distribute within tissues and cells. That could potentially lead to smarter, more targeted dosing strategies for those supplements or drugs.
Interesting. So how's the impact potential rated for this one?
This gets 3 stars,
Yeah.
described as strong mechanistic confirmation,
Meaning?
meaning it's highly valuable because it provides really detailed, high-resolution evidence confirming a known target and mechanism using a groundbreaking new technique. That spatial info is super useful for guiding drug design.
But it's not uncovering a totally new mechanism.
Exactly. It's confirming and detailing a known story about CD38 and NAD plus A, rather than revealing a completely novel aspect of aging. So, strong confirmation, important work, but maybe not a brand new frontier in itself.
Got it. Okay, finally, let's hit another classic hallmark of aging, telomeres.
Right, the last paper for our dive is Telomeric Antisense Oligonucleotides Reduce Premature Aging Phenotypes in Telomerase Mutant Zebrafish. This was posted on bioRxiv May 27, 2025, from authors like Alavina and Rosiello at the Institute for Research on Cancer and Aging of NICE.
Telomeres. The caps on our chromosomes, shortening and dysfunction are definitely linked to aging. What did they investigate here?
They use zebrafish that have mutations in telomerase. That's the enzyme that normally maintains telomere length. So these fish have really short telomeres, and as a result, they show severe premature aging symptoms.
Okay, a model of accelerated aging due to telomere problems. What did they do?
They use something called telomeric antisense oligonucleotides, or TSOs. The core finding is that these TSOs could inhibit the DNA damage response that gets triggered by those critically short telomeres. And by doing that, they were able to rescue some of the premature aging phenotypes in these fish.
Rescue aging phenotypes. That sounds potentially quite significant, even if it is in zebrafish again.
Yes, zebrafish. And the results in that specific model are pretty remarkable. They found that just a single dose of these TSOs could rescue phenotypes, like improving survival quite a bit.
A single dose.
Yeah. And even more strikingly, they reported this effect persisted across multiple generations of the fish.
Wow, transgenerational again?
Seems so in this model. And something they really highlight as unprecedented was the restoration of fertility in the adult fish that were treated.
Fertility restoration.
And suggests that just blocking that specific DNA damage signal coming from the dysfunctional telomeres using these TSOs was actually enough to reverse some aspects of premature aging and even bring back reproductive capacity in these fish.
Okay, a single dose, transgenerational effects, fertility restored. I mean, that's really compelling data if you're a zebrafish. Where does the skepticism come in for human relevance?
And this is where the translational gap looks pretty significant, unfortunately. Zebrafish telomere biology and just their overall aging process, it's really quite different from mammals, especially from humans.
How so?
Well, the dynamics of how telomeres shorten, how telomerase is regulated, how telomere problems impact different tissues, it all varies quite a bit between species. So translating this specific oligonucleotide approach, getting the TSOs delivered to the right cells, making sure they're safe, and achieving similar biological effects in a complex mammal, that's a really major hurdle.
A big leap. What next steps do the researchers suggest to try and bridge that gap?
They propose testing these telomeres in human cell models first, specifically using induced pluripotent stem cells, iPSCs, that have been derived from patients who have telomerase mutations or other telomere-related diseases.
So see if the basic mechanism works in human cells.
Exactly. See if blocking the telomeric DNA damage response shows any promise or relevance there. They also suggest checking if this approach could actually induce senescence reversal in human cells that have short telomeres.
Okay, so considering the really exciting findings in the fish model, but that big distance to humans, what's the impact rating here?
This paper gets just 2 stars. However, and the label is high novelty but low translatability.
Sums it up.
Yeah. The findings are genuinely novel, quite fascinating, especially the transgenerational bit in restoring fertility. Those results definitely make you sit up and take notice.
Yeah.
But that biological gulf between zebrafish and humans, particularly for telomere biology and aging complexity, it just makes it a very high-risk bet for direct translation anytime soon. Exciting basic science, for sure, but not an immediate path to human therapies.
Okay, so let's try and bring all this together now. Looking across these 5 papers, what's the overall picture this week?
Right, well, the overall synthesis really lines up with what we said at the start. This week's batch of papers shows some really strong, detailed mechanistic work. It's happening across several key hallmarks of aging. We saw senescence, epigenetics, metabolism, telomeres.
And some cool technical advances, too.
Definitely. Valuable refinements in our understanding and some genuinely exciting new tools like that ICE framework for spotting senescent cells or the spatial mass spec imaging for looking at metabolism in situ.
But There's always a but, isn't there?
But yeah, there isn't really a clear field-redefining breakthrough here. Nothing in these specific papers seems to put us dramatically closer to that goal of adding, say, a decade or more of healthy life to humans.
Right. How do the papers stack up against each other in that context?
Well, the TPE study in humans is the most clinically relevant right now because it uses an existing procedure. But as we discussed, the effects shown so far are incremental and marker-based. The lysosomal epigenetics work in worms. Conceptually, very novel, really interesting with that transgenerational link. But it faces a huge translational hurdle.
Hmm.
The telomere work in zebrafish had some really striking results in the model, but again, that path to humans is very unclear.
And the tools.
The ICE method and the spatial imaging are probably the most immediately useful for research, advancing the field's capabilities.
Hmm.
But they are tools, not therapies.
So what critical gaps are still really apparent when you look at this collection of work?
The sources themselves kind of point them out. There's still a persistent lack of robust validation for many novel findings, especially getting them confirmed in complex mammalian models.
Right. Moving beyond worms and fish.
Exactly. And crucially, there's still a general absence of data demonstrating really significant functional health span improvements or anything close to that target of, say, 10% lifespan extension in mammals coming directly from these specific interventions. At least not yet.
So we're definitely gaining more detailed knowledge, getting better tools.
Absolutely. The foundation is getting stronger.
But the direct path to those really big longevity leaps isn't obvious just from this week's science.
Not from this batch, no. It highlights the hard foundational work that's still ongoing.
It really does. Fascinating analysis, even if it wasn't quite the blockbuster week maybe some were hoping for. It really underscores the nature of scientific progress, often incremental, sometimes tool-driven.
Indeed.
So, as we wrap up this deep dive into the week's papers, maybe here's a final thought for you, the listener, to chew on. Given the impressive technical advances we are seeing, like ICE for better senescence detection, or these amazing spatial imaging methods, and given the intriguing mechanistic discoveries, even if they're initially in simpler organisms, what do you think is the real rate-limiting step right now? What's preventing us from finding those truly order-of-magnitude breakthroughs in mammalian longevity? Is it mostly about needing better models? Do we need entirely different kinds of tools? Have we maybe been focusing on the wrong targets? Or is it something more fundamental about the sheer biological complexity of aging itself that we're perhaps still underestimating?
That's a huge question.
Mm-hmm.
Probably the central question for the whole field.
It really is. Well, thank you for bringing these sources for the deep dive today. We hope this analysis helps you navigate the cutting edge of longevity research this week.